But what I want to get away with today is for you to understand the difference between GM and gene editing and which of these technologies fall within the precision breeding bill. So I'm going to attempt to keep the science quite light in my talk, but there are a few basics that you do need to appreciate beforehand. So we'll start off with DNA. So DNA is the information that we need to make and maintain us. So all that information is stored into every single cell in our bodies, in our plants. And DNA is made up of these building blocks called bases. So we represent these in this cartoon here with these A's, C's, T's and G's. So you can think of this as the alphabet of life. And these are universal across all species. So the A's that are in you are also the same as the A's that are in wheat. And they make up our genetic sequence. So we now have the ability to sequence genomes, which means that we can take DNA from an organism and we can put it through some fancy technology, which you can now read the exact order in which these letters appear throughout the genome. Now, changes in this sequence confer genetic variation. So we've got a really great example of genetic variation in this audience today. So if I was to compare your genome sequence, it would be 99.9% identical. But those differences in that genome sequence, so that order in which those letters appear, give us the variation that we see today. So if we take something like eye colour, the difference between brown eyes and blue eyes is just a single letter change, so a point mutation. And those mutations are occurring all the time. So mutations can occur through damage to DNA, so we can lose genetic sequence, or we can create breaks in the DNA, and a cell will try to repair those breaks. And in doing so, we can lose the odd base, or we can recruit the different base. So you're introducing point mutations. And this is what plant breeders are looking for. So they're looking for differences within populations to try and identify new desirable traits. So that underpins crop improvement. The other concept is that we inherit half of our genetic information from both parents. So if we take these two different crops here, so they're both the same species, so broccoli, and what you can see next to it is wild Brassica oleracea. So that's what our broccoli would look like if plant breeders had never got to it. But if we go back to these sort of older populations, we've got this huge array of genetic variation and genetic diversity. So imagine we wanted to reintroduce a disease resistance gene from this wild Brassica oleracea back into broccoli. We would cross the two parents together in order to introduce that new trait, that disease resistance. But as well as introducing that gene for disease resistance, we're also going to be introducing tens of thousands of genes that we don't want. So what we'd then have to do is take the offspring and back cross it to the elite line over several generations so that we get something that looks more like your broccoli but has introduced your disease resistance trait. Now, GM technology, which was developed in the 1980s, allowed us to clone just the gene that we want and introduce it into our elite lines. So you can see that that very much speeds up the breeding process. Now, as I mentioned beforehand, those A's, T's and C's are universal, and because we're no longer needing to sexually cross plants together to introduce these new genetic traits, it means we can now take genes from unrelated species. So I could take a disease resistance gene from wheat and introduce it into broccoli. But I could also take a gene from a completely unrelated species, like a bacteria. And so most of the first generation GM crops that are out there, so the herbicide tolerance and the insect resistance, contain genes from bacteria which confer these traits. So just to define that, when we've introduced DNA or genetic sequence from a completely unrelated, uncrossable species, we refer to these as transgenic, so trans meaning different. And where we've introduced genetic sequence or genes from the same species, we refer to these as cisgenics, and the new precision breeding bill gives scope for cisgenics to be included. So let's give you an example of a GM field trial. So this is work by colleagues at the Sainsbury Laboratory. So what you're looking at here now is a field of potatoes. These have been untreated, so not sprayed by fungicides, and I hope you can see that half of them have been decimated by late potato blight, and half of them look green and healthy, and those of course are the GM crops there. So what's happened here is the group at the Sainsbury Laboratory have isolated 3 genes from a wild potato relative and reintroduced those into those commercial lines. So I hope this gives you a good example of where we can use genetic solutions to reduce our need for chemical inputs. This technology is not being used in the UK, but it is being used in the States and other parts of the world where they've embraced the technology. So gene editing is slightly different. It's a tool. So there's lots of different gene editing approaches we can use. The one that's used most widely by scientists is one called CRISPR. So CRISPR just stands for Clustered Regularly Interspersed Palindromic Repeats, which doesn't help you understand the technology at all. But essentially, it's a tool which allows us to cut the DNA at a precise location. So depending on what you do with this tool will depend on how it's regulated. So in the very simplest of form, if we make 2 cuts to the DNA close together, we can delete genetic sequence. If we make a single cut, we're hoping the plant will then try to repair that cut. It may then recruit the wrong base, or it might delete the wrong base. And so you're introducing a point mutation at that location. So it's doing something that could happen naturally, but we're putting it at a very precise location. And so using this technology in this way, these would fall under the Precision Breeding Bill. We can also use the technology to open up the genome at a precise location in order to introduce new genetic sequence or actually take out a gene and replace it with a gene. We can consider this to be the upgrade to GM technology. It will still be regulated as GMOs if that new genetic sequence comes from a different species, but if it comes from the same species, this would be a cisgenic and it could fall under the Precision Breeding Bill. So I want to give you a worked example of that. So here we've got a field of oilseed rape. Now our farmers face great challenges even getting to this stage at the moment, but imagine you've got a really good field of oilseed rape. What you don't want to do is then lose some of your yield as a result of early pod shatter in the field. So can we use gene editing to address this problem? So what I'm showing you now on this scary looking slide is a page of genomic sequence. If I was to show you the entire genome sequence for oilseed rape, I'd need to show you about 1 million screenshots like this. So all that information is packed into our cells. We're now at this sort of big data time. We can compare genomes between species and within species to enable us to identify our gene sequence. So this is our candidate gene. This is the gene that we believe is involved in pod formation. And then we use computer software. So again, this is all happening on a computer. We use computer software to identify a 20-base sequence that is unique to your gene of interest and nowhere else within the genome. We would then design a molecular guide that we can introduce into the cell that hunts out this sequence. So you can imagine it's a bit like a Google search engine. We're hunting for the sequence that we want to make a difference to. We then introduce a nuclease, so an enzyme which cuts DNA. So we can think of that as like a pair of scissors. So we're cutting our gene at a precise location. So we're not adding anything new, we're just making a break in that region. And this is what it looks like. So here what you're seeing now is a cross-section of 2 pods. And you can see where that green arrow is, that's where our valve margin would form. So that's where your pods would break open. And you can see where we've targeted our guide, and where it's circled is where we've made that break. And you can see that we've got a C to T change and we've lost a G base. So in the billions of letters in the oilseed rape genome, we've made 2 changes, and hopefully you can see in the edited version that valve margin no longer forms. So that's how we've disrupted that gene from forming. So that's a very flash view of the difference between GM and gene editing. And so gene editing and precision breeding really has the potential to benefit farmers, the environment and the economy. And there's a whole host of different things that we can do with it. But the ones that are coming through to market currently or in the field trial stages are work to develop more resilient crops. So we're seeing some nice work looking at disease resistance. You can just about make out on that far left, is it left? Right-hand side image, some pictures of some wheat. And here, so wheat contains something around between 100 to 200,000 genes. We've made 2 deletions in 2 genes, and we've got yellow rust resistance. We're also seeing some work to develop pest-resistant plants, as well as plants that are better adapted to climate change. So there's some nice work going on in Brazil. So we've got some drought-tolerant soybean that has been brought to market. And we can also use this technology to open up new markets, so to create new crops or to develop underutilised crops that are better adapted to growing here. So in this one image here, we've got cowpeas, and they've been edited so that, so that's a crop that is hand harvested. These have been edited to change the plant architecture so they lift higher up off the ground so they can be mechanically farmed. We're also seeing work to increase yields. On this top image here, you can see some wheat. And in this particular piece of work, they've made 3 deletions to 3 genes, and you can see an increase in yields of up to 20%. Also work to reduce food waste as well. And the other things we're seeing is editing to make healthier crops. So in the US, we've already got these salad greens, which are being edited to have higher nutrient levels. And at the John Innes Centre, we've got these vitamin D enhanced tomatoes. So that's a very quick flash talk through, but I hope I've managed to convince you that the new Precision Breeding Technologies Act really gives us this opportunity to take advantage of our knowledge of genomics. So our ability to sequence genomes is fast improving. When you think that the first human genome took 10 years to sequence, cost billions of pounds, we could now sequence a genome within a day. So this technology is rapidly advancing. So it very much brings us into this new era of intelligent precision-based breeding. The new Act brings us in line with the views of a number of other countries across the globe. And so, you know, it gives us the opportunity to stay competitive if we're allowed to embrace it. But I think what's been quite clear in the talks today is that, you know, science is only part of the story. So I can tell you what's possible and that the technology is safe, but it's really down to consumers and industry, so the people in this room, as to whether these technologies will be embraced.